everyone. My name is James Smith, and my presentation is on the role of surface chemistry in polyethylenimine grafting and the removal of chromium-6 by activated carbons from cashew nutshells. Here we go. So the first thing I want to go over is what an activated carbon is. It's a porous and amorphous structure as seen in the figure on the right. There are different sizes of pores such as macropores and micropores, as seen here with the laser pointer. Activated carbons are also known for having a high number of surface functional groups, which is seen in this figure on the left. Most importantly for our research are the lactone groups, but there are also groups such as carboxyl, carbonyl, ether, phenol, just to name a few. We use cashew nut shells in our research. So this is just a picture of a cashew apple. This is the entire plant, which is the apple, and then a nut on the bottom. And then obviously we use the shell around it. And one more thing I'd like to mention is polyethylenimine. It's a cationic polymer, meaning it has a positive charge, and it has been shown in literature that adding it onto activated carbons can increase their removal efficiency. So we tried that here as well. So chromium exists in two forms, chromium-3 and chromium-6. Chromium-6 is toxic, and it is known to cause cancer, respiratory problems, and stomach ulcers. It is introduced into wastewater from industrial manufacturers such as dye and alloy manufacture. So the way that we looked at this in the lab is that we took cashew nut shells and activated them with phosphoric acid. We carbonized them at 400 degrees Celsius under nitrogen gas and labeled them CNP. CNP stands for cashew nut and phosphoric acid. Then some of our samples had PEI impregnation. PEI is polyethylenimine, and those were labeled CNP-PEI. For our adsorption studies, our carbon samples were treated with chromium-6 solutions in a range of 10 to 200 parts per million. They were equilibrated overnight on a shaker, and then the filtrate was measured for its absorbance with UV vis. For pH studies, we did the same as adsorption studies, except we lowered the pH beforehand with 0.0 molar hydrochloric acid. And then we used FTIR and XPS in order to analyze the carbon surface for its functional groups. The first part of the data I'll go over is how we fitted it to the Langmuir model. So the Langmuir model assumes that there is a homogeneous monolayer adsorption happening. So our results are in the highlighted light blue color. And these tables are just showing for activated carbon and activated carbon with PEI with different literature values. So QM, which is the maximum absorption capacity, is in milligrams of chromium-6 per gram of carbon. So as you can see, overall, our values are essentially the same for CNP and CNP-PEI, and they're better than most of the QM values reported in other literature. For pH studies, the main point I want to point out here is that at pH 2, we see the maximum removal efficiency for both CNP and CNP-PEI. As it goes up towards 5 and 5.5, we see the pH, the removal efficiency sharply decreases. This just shows us that pH 2 is the optimal pH for our carbons. So for FTIR, I'll just briefly go over how to read this graph. We have CNP-PEI and CNP-PEIE, and then CNP and CNPE. The E stands for exhausted. So these are the samples after chromium-6 is removed. So the first groups I'd like to point out are the 1115, 1020, and 1430, which are right here on the PEI. This corresponds to CN stretching, and these appear after PEI grafting, which shows us that the PEI grafting procedure worked. This is not present in CNP, which is good because the polyethylenimine is the part that's adding nitrogens. And then these groups actually decrease between the initial and the exhausted, which shows us that they are part of the removal process of chromium-6. Then for 1400 and 1000, which is around this region here, there are many different functional groups that overlapped, but the most important for us were the CO stretching and phenols. So the CO groups decrease between the initial and exhausted for both, which shows us that they are active in the removal process. And then 3300, 1690, and 1580, they're marked around here, correspond to OH and C double bond O's, and they form after the removal of chromium-6, and they participate in the process. So just for example, right here at this 3300, you can see in the initial it's flat, but then the intensity moves here. So that shows us that OHs are appearing on the surface. So for our XPS data, I'd like you to focus down here on our mechanism while I talk about it. So this shows our data for CNP and CNP with PEI. So this mechanism, the first one, is for CNP at pH 2, which is the optimal pH as I mentioned earlier. Here you can see our lactone groups, and they undergo acid catalyzed hydrolysis in order to form phenol groups right here. Chromium is in the form of anionic hydrogen chromate, which is attracted to the positive carbon surface. This reduces chromium 6 to chromium 3. This is seen in our XPS data where only chromium 3 is present and there is no chromium 6. As this reduction is occurring, the phenol groups are oxidized to carboxyl and carbonyl groups. So between here and here, you can see that happen. And this is backed up by our table. If you look right up here, you'll see our COs from phenols decrease before and after, and then our carbonyl and carboxyl groups are increasing. Then this mechanism is for CNP-PEI at pH 2. As you can see, the amide groups are undergoing acid hydrolysis to form carboxyl groups in their place. This is supported by our N1S data where the number of amine/amide groups is decreasing after absorption of chromium 6. And since only chromium 3 is present on the surface, again, this tells us that complexation of chromium 6 with the amide groups isn't happening, and that instead we believe it's happening in this mechanism, which is very similar to the CNP one, where you see that CNP is reduced to chromium 3 again, or sorry, chromium 6 is reduced again, and that the phenol groups are oxidized to form carboxyl and carbonyl groups again. And this is also shown in the table right up here as it was for CNP. So for our conclusions, the Langmuir model was used to measure our maximum absorption capacity, which was 340 and 320 milligrams per gram for CNP and CNP-PEI respectively. PEI did not increase the capacity since these were essentially the same. FTIR and XPS data confirmed that our PEI was successfully grafted and that COOH and C double bond O groups are active in their removal process of chromium 6. And since CNP and CNP-PEI have the same capacity, we figured that chromium 6 is reduced to chromium 3 during the removal process rather than having it complex out of solution. And our phenol groups are oxidized to carboxyl and carbonyl groups. And these are just my acknowledgements. Thank you.